Good afternoon, everyone. Pleasure to welcome all of you to our Wednesday afternoon lecture. And I think it's fair to say you're in for a treat of an inspiring presentation from Dr. Julie McConney, who will be talking to us about sickle cell disease from her perspective as a leading researcher on that condition in Tanzania. My own first research project as a postdoctoral fellow 30 years ago was on hemoglobin. Many of the people who got involved in human molecular genetics started in that particular system because of its elegance and what we hoped would be its simplicity, although that was probably a hope that wasn't quite to be realized. But we recognized early on that the amount of fetal hemoglobin produced by somebody with sickle cell disease had a very significant effect on how severe the disease was going to be. And so much of the efforts over the last 30 years have been to understand that nature of hemoglobin production of the fetal sort and to try to figure out how to utilize that in some way that would be therapeutically beneficial. And some significant progress has been made, as you will hear from our presenter. But we still have a challenge in front of us. Just 3 years ago, we celebrated the 100th anniversary of the first paper describing sickle cell disease. So it's been more than a century, and we still have only one approved drug for the treatment of this disease. Lots of efforts here at NIH and across the world to try to change that. I think, again, with a sense now of considerable momentum coming from advances in the way in which we're understanding the molecular basis of this disorder. But of course, the main burden of this disorder is not here in the United States. It is in sub-Saharan Africa, where in fact, as we all know and learned in basic biology, the influence of the malarial parasite has resulted in a substantial increase in the allele frequency for the sickle mutation. I'm told that in Tanzania, where Professor McConney spends most of her time, 8,000 to 11,000 children are born every year with sickle cell disease. The majority don't know they have it, and without interventions, up to 90% of them are not likely to survive into adulthood. So this is an enormous challenge and a responsibility, I think, for all of us who care about the health of the world to address. And it is in that context that I think we're particularly fortunate to see emerging a figure like Julie McConney in Tanzania, determined to bring all of the tools of molecular biology and therapeutics to this disease, but in the context of the place where the disease carries its greatest consequences. And so it is a joy to be able to invite her for this week, and she's been visiting with quite a few people over the course of the last couple of days to talk about ways in which those dreams of a better time for sickle cell disease could come true in Africa, and of course here in the US as well, and in other countries across the world. Dr. McConney got her MD degree from the University of Dar es Salaam in Tanzania, followed that up with training in internal medicine at the Royal Postgraduate Medical School in London, so she's a British trained physician. More recently, she obtained a sickle cell disease PhD from the Open University in the United Kingdom, so she's an MD PhD. She's been recognized for her work in several ways, by being supported by the Wellcome Trust. And I heard about Julie from several people at the Wellcome Trust who are really excited about the work that she's doing. She also has been, just last year, chosen as the outstanding scientist in Tanzania by the National Institute of Medical Research in that country. And she received the year before that the Royal Society Pfizer Award from the Royal Society of the UK. So I think you're in for a treat to hear this perspective about this original molecular disease from someone who sees a lot of it in her own country in East Africa and who has a vision about how we might collectively conquer this disease once and for all. So please join me in welcoming to the podium Dr. Julie McConney.
Thank you very much for inviting me to share with you our experience in working in sickle cell disease in Africa and how we think we can work together to make a difference as far as healthcare is concerned, as well as contribute to science in Africa and globally. So the burden of disease is high in Africa. This was a map showing the global frequency of the S gene that was published in 2012 by the MAP group from Oxford. And what it shows is that the allele frequency is highest in these areas in sub-Saharan Africa. And using geostatistical methods, they've estimated that over 300,000 children are born with SS disease every year in the world. Of these 300,000, 240, almost 76% of them are born in sub-Saharan Africa. So the burden of disease is greatest in Africa. Unfortunately, if you're a child and you're born in sub-Saharan Africa, the chances are nobody will know that you have sickle cell disease. And therefore, for those children, if they're very fortunate, 50% of them will survive to adulthood. If not, more than 90% of them will die before the age of 18. Whereas if they had been born in the UK or in the US, the chances are 98 or over 90% of them would have survived to the age of 18. So this is something that is a tragedy, in that it is something that we can do about with the few tools that we have available. And this is demonstrated by this report that was published by Oyani and et al. This is a group from the CDC, which looked at the trends in mortality among sickle cell patients in the US. And what it showed is that amongst the 0 to 3-year-old age group, there's been a significant reduction in mortality. And this is thought to be associated or thought to be due to introduction of newborn screening and prevention of infection. And they've been able to achieve, or you have been able to achieve here in this country, a huge reduction in mortality. The thing that is worth noting is that the mortality in the older age groups, unfortunately, has not changed during this period of time. So despite the achievements that have been reached, there is still quite a lot that needs to be done amongst adults or amongst older children. Unfortunately, none of these interventions are available in Africa. We don't have newborn screening. We don't have dedicated prevention of infection measures. We certainly have few comprehensive care centers, and very few patients are on hydroxyurea. However, with efforts from a few individuals within Africa working in various countries, there has been quite a lot of advances in the past 15 years. I think the leading work that has been done by Kof, who is based in Philadelphia, but he's from Ghana, has really been an inspiration to a lot of us working in Africa to see that what can be done in sickle cell disease with the limited resources and with the little attention that is paid as far as sickle cell disease is concerned. And they have been working on setting up newborn screening as well as establishing comprehensive care centers in collaboration with the Brazil group. In Tanzania, we started a pilot project as well, learning from what is being done in Ghana, Benin, DRC, and a few other countries, that it is possible to do newborn screening. And last year in November, they introduced pneumococcal vaccine as part of the expanded program of immunization. And for us, this is very exciting because it does mean that healthcare measures will start coming into place and we'll be able at least to reduce mortality in the first 3 years of life amongst children. The effect of that is that with the reduction in mortality, we will definitely see, obviously, increased survival. And what we did is we estimated that with a reduction in under 5 mortality, this is without doing newborn screening, without preventing infection, you will automatically see an increase in the proportion of children who will survive to adulthood. And this is because they have better access to childcare, better access to vaccines, and you will see an increase in the number of children who obviously are born, but an increase in the number of children with sickle cell, which is this line that shows here, So as Dr. Collins says, at the moment, we estimate that around 11,000 children are born with SS disease, but these numbers are bound to go up, and we need to think about what are the things that we're going to do to make sure that the children who survive have a good quality of life and have a longer life expectancy than they currently do. So I just wanted to demonstrate the fact that this is a chronic disease. Once we're able to achieve this reduction in mortality in Africa, then we still need to think about how we're going to improve the quality of life in the older children and how we're going to make sure that they have long, productive lives. So there's been work, unfortunately, it's in a few centers. So this is the pediatric complex within the hospital that I work in, which is a public national hospital. But there's work, and we're working to provide regular clinical care, trying to promptly diagnose and treat infections, provide blood transfusion, hydroxyurea, and we've been discussing with people in the US, including here, as to whether it would be possible to start thinking about at least preparing patients for stem cell transplant, because this is something that some patients are able to consider and would benefit from having this as a treatment therapy. The thing about sickle cell disease is that although it affects, or it's primarily a disease of the red cell, it has an impact on almost all organs within the body. The primary thing that causes a lot of morbidity is pain. Patients come in with severe debilitating pain, but then you also have other complications, stroke, anemia, chest infections, and as well as acute chest syndrome. Unfortunately, despite the fact that it was described over 100 years ago, we don't know very well what happens as far as the anemia is concerned, what causes it, what's the spectrum of anemia, what are the different etiological factors, in order for us to identify what are the right interventions that we need to put in place. So as far as interventions are concerned, there's been a lot of work over the past 50 to 60 years to try and look at agents that could be used to treat sickle cell disease, and they've been targeting different known pathological mechanisms. So some have been looking at reducing hemoglobin S polymerization, others have been looking at how they can reduce the vasoocclusion, restore nitric oxide, and all of these are things that are being tried in different centers within the US and other countries within the world. Unfortunately, despite all of this, hydroxyurea is the only drug that is licensed for use in sickle cell disease. So this is a condition that has been around for over 100 years, and yet we have only one drug that is available for use. When you compare it to all the other leukemias that we see, you compare it to conditions like HIV, they've been discovered more recently, and they have more than 5, maybe in some instances over 10 drugs are available. And yet in sickle cell disease, only one drug is available. So just to summarize, this is a disease, it's a genetic disease, has a high burden of disease, has high prevalence, high morbidity and mortality, and there's an unmet medical need. And we only have one drug that is available. Now, we can look at this and say, well, it's too much is involved. If we haven't been able to solve it in 100 years, what are the chances that we'll be able to solve it within our lifetime? And the answer is really from these 3 things, which are identical to the characteristics of sickle cell disease that I've just summarized. This makes sickle cell disease a model disease for genetic research. Alchala suggested that if we want to study a genetic disease, we really need to look for one that has higher heritability, has an unmet medical need, and has limited interventions, which means that sickle cell disease is a model disease for genetic studies. And within the grand challenges that were discussed after the completion of the Human Genome Project, I'm particularly interested in how we can translate genomics to health, mainly because sickle cell disease has such a huge burden, and if we're able to solve sickle cell disease using genomics methods, then it means that we will be able to achieve this grand challenge. And if you're able to do it in sickle cell disease, it means that we will be able to learn and use the tools in other conditions that have a more multiple, a more complex genetic contribution. So this is our kind of vision to say, how can we look at genes, how can we understand how it causes disease and make children with sickle cell disease healthier and survive and become adults, looking at this in the context of all the social and cultural factors that contribute to disease variability, as well as the environmental factors that influence the spectrum of disease. Now, we've had to, I'm sure, as this is probably the case with a lot of people working in science, is that we have to convince the healthcare or the clinicians why we need to do research. And I think this paper that was published by Stuart Orkin's group demonstrate the fact that with genomic approaches, we can not only ameliorate the disease, but we can actually solve some of the issues and find possibility, if possible, the cure for sickle cell disease. So just summarizing the state of sickle cell disease in Africa, these are the kind of discussions that we have and the debates that we have and the things that we need to answer. As far as healthcare is concerned, the question is, well, why should we focus on a genetic disease when the state of the healthcare system won't be able to provide care? There's limited human resource, and what really is the point? And this is something that is very difficult to deal with when people say that, most sickle cell patients die in childhood. There's no point working on them. And this is something, and this is a perception that we absolutely have to change. Within the scientific community, the things that we have had to deal with is to say that, well, why do we need to work on sickle cell disease? We have all the answers. We know that fetal hemoglobin ameliorates disease. We know that everything that there is to know about sickle cell disease. So why on earth should we invest in genomic research to do anything else? Why should we repeat the studies that have been done elsewhere? The answer is really with these facts that 100 years ago, over 100 years ago, there was the first clinical description. The molecular basis of it was more than 50 years ago, and hydroxyurea is the only drug available, and we're not even talking about a cure for sickle cell disease. In addition to sickle cell, this is something that I thought was relevant or pertinent, and it's intriguing for us coming from an area where we see so many people with trait, but this is a big discussion within your country about what are the issues, or how do you deal with the reports where you have young, healthy athletes suddenly dying and after severe, exercising vigorously, and they're found to have sickle cell trait. And here, as Africa, we can contribute towards this debate by saying, well, here's a chance for you to look at what is the physiological response to exercise in individuals with sickle cell trait, so that we can at least inform this decision and make an informed choice as to what should be done within the USA. So how can we contribute to this debate? Not just the sickle cell trait NCAA one, but the more general debate. I'm going to briefly try and say what is the public health justification for working on sickle cell disease and using genomic approaches, the scientific justification, what are the gaps in knowledge, what are the things that maybe we should focus on, and what are the resources that are actually available. We're not starting from scratch. And this is just trying to demonstrate the 4 key areas when looking at genomic research and trying to look at how we can make health benefits with the phenotyping, genotyping, understanding how genes function, as well as translation of the knowledge that we generate here into health benefits. And there are lots of other things that are cross-cutting in this whole area. So the first thing is really the phenotype, and I think this is really the basis of a lot of the genetic association studies that we need to do, in that there is no point having sophisticated genotyping capabilities if you haven't got a well-characterized phenotype in place. And so being able to phenotype individuals, being able to characterize the type of stroke that patients have, the type of hemoglobin hemoglobin F levels that they have is really, really critical. The advantage of this is that, or the thing about this is that all of these are techniques that are available in Africa. Secondly, by doing or by carefully characterizing the phenotype, you're doing something for healthcare as well, because all clinicians need to make an accurate diagnosis so that we can know what it is that we need as far as treatment is concerned. So here you're able to do both, to do something that has both a scientific outcome as well as a healthcare outcome. As far as interventions are concerned, as I said, we will be able to introduce the interventions that work, but as you can see here, it reduces mortality, but it doesn't reduce it to zero. So what are the things that make some individuals, despite the fact that they're on penicillin prophylaxis, they receive pneumococcal vaccine, they're still susceptible to infections? And what do we do to change this, to reduce this even further, so that to make sure that people have long, productive lives? As far as sequencing is concerned, the strategy that we have taken within the consortium of sickle cell disease that we've been working on is to say that as far as sequencing capabilities are concerned, there's enough of that outside, whether it's in Sanger, it's here in the US, it's in Beijing, or it's in South Africa. There is capacity, and strategically, for now, it makes sense for us to outsource this to be done in places where there's a lot more expertise in being able to do that. But whilst all of this is being done outside, we need to be able to empower individuals to understand that it's important to look at the human genome variation within Africa. We need to understand what is the genetic variation in order to interpret our results properly, and in order for us to be able to make sense of the information that we're able to generate. As far as informatics and analysis, I think this is a critical area for geneticists, particularly, or people working in genomics, particularly in Africa. This is an area that is worth investing in, because this is something that can be done. You know, you have all the data being generated outside of Africa, but this is something that can be shared. There's a lot of informatics and analytical capacity that is there globally, and this is something that we could use. And because this is an area that we thought was strategic in terms of investing in, we worked with the NHGRI as part of the H3 Africa initiative, and there is a consortium of informatics and analysis that has been formed across Africa, and this is called H3A Bionet. And what we're trying to do is to say that we have capacity. It's very limited, but we have individuals who have the capacity to handle data, to analyze the data, but they're all scattered around Africa. And it doesn't make sense for each African country to try and build up its own capacity, especially initially alone. So it makes sense if we all work together as, you know, in a Pan-African kind of way, and push this capacity up. And so this is a network that has been funded again by the H3 Africa initiative, and it's led by Nicola Mulder from South Africa, and it involves, as you can see, a really good representation of nodes and associate nodes across Africa. And the aim, so specifically for sickle cell disease, the aim is currently, if one was to use this as a model, to say, well, we collect the genetic data that is genotyped, whether this is in the US, in the UK, or outside of Africa. This data is generated there. There is an informatics platform that data is uploaded to, and then we have data that's being collected in centers in Africa. And this is phenotype data, as well as data that looks at all the environmental factors that would influence disease. And then you have platforms that you're able to look and work collaboratively with scientists anywhere really in the world to look at this, so that we can have a collective approach to solving this problem. As far as environmental factors, again, you know, the whole phrase of Africa is not really a country. There's huge heterogeneity as far as environmental diversity, looking at things like socioeconomic status, looking at things like access to healthcare, looking at things like climate, looking at things that, you know, just the education levels that we have. And all of these influence the spectrum of disease. They influence the genetic contribution to disease, and they influence how patients fare. And all of these are things that have to be taken into account to be able to understand what exactly happens in patients with sickle cell disease, and what are the things that we should be looking at in order to understand why some patients do much better than others. And the thing about sickle cell disease, despite the fact that it's a known mutation, but it's very different. So within Africa, there have been 3, sometimes 4, you could say, haplotypes that have been described. And this is a description that was done more than 20 years ago. And now we would be in a position to really sequence a lot of the populations here so that we can, first of all, validate whether this is correct, and then be able to look at all the other genetic factors that would contribute to the distribution, which is very marked. And this is important because if you have sickle cell disease with a Bantu or Central African Republic haplotype, your chances of having severe disease are higher than if you have the Senegal haplotype. And then there are other factors as well that come into play, you know, the distribution of malaria, the distribution of infections, the serotypes of bacterial infections is different. And so these are things that, again, have to be taken into account, and one can't assume that sickle cell disease in West Africa or in London or in Boston is the same. And we have, with all the access to the genetic capabilities that we have, there have been a lot of reports that have said, okay, what are the factors or what are the genetic factors that are associated with stroke, associated with gallstones? And there has been a huge number of literature and reports that have done this. This is great, and this is really showing that this is something that can be done. The limitation is that it has been done in African American populations, in Jamaican populations, in European populations, and they're small numbers and they have considerable admixture. We are able to, or we're in a position now as a scientific community to validate a lot of this work in African populations which are homogeneous that will give us a bit more information. But then one could say, well, why should we just repeat studies that have been done elsewhere? And I think this demonstrates the need of investigating this again in African populations. So these are 3 genetic loci that are associated with fetal hemoglobin. We know that fetal hemoglobin is genetically determined, and we know that high levels of HbF are associated with mild levels of disease. So when you look at sickle cell populations outside in Africa, in the US, in Europe and elsewhere, this contributes to about 20% of hemoglobin variability. When we repeated these studies just to really just characterize the prevalence and the association in Tanzania, we found that the contribution or the prevalence of this is much lower, suggesting that, one, there's still quite a lot that we don't know, and what we know from populations outside of Africa may not really or necessarily apply to the sickle cell population in Africa. So we do need to work together to do this. As far as hydroxyurea is concerned, which is the drug that is available, we should start using it. And I think it's really important not to delay treatment because this is something that does make a difference. It reduces pain, it reduces mortality. There really shouldn't be a debate as to whether it should be used in Africa or not. However, we don't know how it works. So by introducing it in Africa and conducting interventional trials in Africa, what we will be in a position to do is conduct studies to say, well, how does it really work in addition to, other than increasing fetal hemoglobin? How does it affect the vascular endothelium? How does it affect other factors as well? So again, doing this in a way where you're introducing an intervention whilst you're understanding how drugs work and how people respond to it is something that can be done so that we're achieving both things, achieving a scientific objective as well as improving health. And so we just come back to this. It's a huge, I haven't talked about functional genomics, I haven't talked about translation as such, but what I wanted to show is that it's something that is ongoing and there is a lot that can be done with the tools that we currently have. What I wanted to demonstrate here is that it will require quite a lot of people, and we know that within Africa there are limited numbers of individuals with the capabilities of being part of this whole program. So we do need to think about education and training, and we need to think about education and training as far as healthcare is concerned first and foremost, because we have to improve health amongst children with sickle cell and individuals with sickle cell disease. So we need to train doctors, nurses, healthcare providers, medical technologists so that we can improve diagnosis and improve treatment with the limited tools that we have. We need to train scientists in all the different areas, make sure that we know what are the right ways of communicating complex, the complexities of sickle cell disease, genetic disease and research, and also improve genetic literacy amongst policymakers. And so we have to have formal training programs, short courses that complement these formal programs. And this is where, again, the capacity that we're talking about or the programs that we're talking about extend beyond sickle cell disease and beyond genomics. And so the real urge or the real message that I'd like to say is that we really need to think about an integrated approach where we think about training so that we have biomedical scientists and people. We think about healthcare whilst we're doing research, and we think about advocacy at different levels, being able to communicate properly with our patients, with the public, as well as being able to convince policymakers, convince people who make decisions that it is important to address this condition. So there are clinical research centers. Again, this is in Tanzania, but we're having them across Africa. People working on sickle cell disease in their own way with limited resources, but they're amazing activities that are ongoing within Africa. We have formed regional networks because similar to the H3A BioNet, it doesn't make sense for everybody to work in isolation. There's strength in working together. So this is a regional network that has brought together countries in East and Central Africa, and the thing about those countries is that, you know, we have a similar haplotype, but also similar other environmental conditions, so it makes sense for us to work together. So this is a group that was formed and is being led by Leon Shilolo, who's based in the Democratic Republic of Congo, and meets annually to really think about what are the research questions that are critical and what are the healthcare issues that we can advocate or push our policymakers to say that we do need to introduce screening. And then this is a consortium. So Sickle Charter stands for the Consortium of Health, Advocacy, Research and Training in Africa. And it's a consortium that is pan-African, as you can see. At the moment, we have 5 countries that are part of that consortium, but we're part of the smaller network that I've showed, the regional network, and part of the bigger network that I'll show you in the next slide. And this is, these are countries these 5 countries: Tanzania, DRC, Cameroon, Ghana, and Nigeria. And what we've done is we've come together and we say, well, what are the strengths that we have and what are the questions that we need to approach whilst we are also improving or developing healthcare within our country. So Ghana has been able to start universal newborn screening in Africa using health insurance funding and resources, and this is something that we're all working on, improving healthcare as well as improving the scientific questions in our scientific capacity. And we're all part of a global sickle cell disease network. So this is a network that has 23 countries, but it's inclusive, so this number is probably much higher than this. And this is is a network that was started by a group in Canada, by Isaac Odame, and the aim is really, again, at global level. We know that if you look at the number of people working in sickle cell disease compared to oncology, it's very small. So again, let's all work together, learn from each other, and see how we can push health and research in sickle cell disease so that we can make a difference. So finally, I guess it's really, when you look at this, I think this is the final couple of slides, the final slides. It's really why I think that we should think about genomics. And the reason why I feel that we should think about genomics in the context of sickle cell disease is because when you think about, or when you talk about sickle cell disease, you will immediately have people disengaging, either because they would assume that, well, this is a red cell disorder, or this is a disorder that only happens in Africa, or this is a disorder that is not really something that is of interest of me. But if we say that this is a model for genomics, and genomics is something that it doesn't really matter as far as phenotyping is concerned, if you're a cardiologist, it will be of importance. If you're, if you're a gastroenterologist, this is something that is important. So as part of the whole thing of promoting genomic research in Africa, Charles Rotimi and the group that formed the African Society of Human Genomics have been really instrumental in kind of advising and advocating for investment in genomic research in Africa in collaboration with partners all around the world. And this led to the Human Hereditary and Health in Africa that was launched a couple of years ago and has already funded 5 projects, disease-specific projects, as well as capacity in strengthening bioinformatics and biorepository. So these are the diseases that they've funded. This is a huge, it's considerable investment, and it will be able to initiate a different way of working in health, a different way of working in science, and a different way of collaborating across the world. So finally, just wanting to emphasize the point that there is a lot capacity, a lot of resources that are available as far as science and technologies is concerned. And it really is a pledge to say, how can we use all this technology that is currently available, that we have access to make a difference as far as health is concerned? It's something that we can do if we use the technology to understand this and make a difference in terms of healthcare. So my final slide really is to just say we really have unprecedented opportunity with all the tools that we have in hand, with all the capacity and with all the resources that we have to make a huge difference as far as healthcare is concerned for a genetic disease. Through clinical research centers, we will automatically improve the health of individuals. We will be able to utilize genomic tools to cure a disease. So we should think about curing sickle cell disease as opposed to looking for ways of finding drugs to ameliorate the disease, but let's just think about curing the disease. And once we do that and we use genomic tools, then these tools are not just limited to sickle cell disease. They can be used for all other conditions, because if we're able to understand sickle cell disease, it means that we will be able to understand, or at least try to understand or contribute to the knowledge to understand more complex diseases. So with that, I'd like to thank you for your attention, and thank you very much for inviting me to share with you what it is. And thank you to all the families, staff, and people that we work with, and the funders. Thank you.
Thanks, Julie, for that presentation. We have time for questions, and there are microphones in the aisles, so please, if you have a question, take advantage of that so people listening over the web can hear. Yes, please.
Thank you, Dr. Makani. It's very interesting. If you would, please, the young, healthy athletes with sickle cell trait, could you describe the pathophysiology a bit, how that works?
Okay.
Yeah.
So do you want to on? Okay, so I don't think it's known, but I think the hypothesis is that with extreme exercise, there is deoxygenation, and this causes the sickling process to happen, and this will result or may result in pathological disease. But I don't know whether somebody know. But this is the whole debate, I think, is more to do with the fact that, was this an incidental finding in the athletes who were reported to have disease, or is this a real disease? Was the sickling that was found the cause of death, or was it just an incident? And I think the thinking is that this is more of an incidental finding rather than a cause and effect.
It's interesting because I think a lot of folks may be under the impression that the trait is benign.
That's right.
Relatively. And in general, we have always said that, yeah. And there is this question whether under extreme circumstances, for instance, depressurized high-altitude flight or extreme athletic activities, whether in fact there might be a phenotype that would be induced for a sickle cell trait carrier. The athletic debate continues to be a very controversial one, despite the NCAA's decision to suggest that screening should be done. It's an active discussion. I think that's fair to say. Roger.
Yeah, Julie, lovely presentation. Thank you. I have a question about the epidemiology and mortality from sickle. It's clear that in Central Africa, as you've outlined in those maps, sicklers have a predisposition. They have a survival advantage, perhaps because of the malaria. At the same time, my understanding is that 90% or a high percentage of children die before reproductive age. So there's a harvesting of children before they can be enriched in the population. How do you account for those balancing effects of increased survival from malaria but increased mortality from childhood infections and lack of care?
I think this is why sickle is used as an example of balanced polymorphism because of precisely that. With increased mortality, if you have the SS gene, this is balanced out by increased survival if you have the AS gene. So the gene will, has reached equilibrium and it's high enough so that you will have a lot of people with the AS genotype, and that means that you will still be having children with SS disease.
You mentioned the importance of establishing these centers of excellence, and I wanted you to think forward a little bit in terms of what that might make possible. You also showed a slide about the work that's going on in terms of trying to understand the role of this gene BCL11A and its protein product in regulating fetal hemoglobin. And there's a lot of excitement about the idea that antagonists to BCL11A might turn out to be wonderful therapeutics for sickle cell disease because they would turn on fetal globin in people who would benefit from that. So imagine that, in fact, that enterprise gets going and gets to the point of having a possible drug candidate. There are questions will be raised, well, where should these clinical trials be conducted? Can you imagine conducting that kind of clinical trial, at least in parallel with what might go on in a place like the US, to mount such trials in Africa? What would be the challenges there in terms of logistics, in terms of ethical concerns? You must have thought a bit about this and hoped that that day would come soon.
Yes. So fortunately, we won't be starting from scratch. So there are clinical trials that are ongoing with HIV, malaria vaccines, and so the infrastructure or sorting out the infrastructure for clinical trials is already in place. So there's expertise in terms of getting informed consent, there's expertise in terms of being able to recruit patients, follow them up, that is there. So when we go in, rather than say we're starting from scratch, we will be able to look to our colleagues who are working in these areas, who've done the clinical trials, and say, well, what are the issues that we need to do, and can we use the same platforms or the same infrastructure to do the trials that we have. There will be other issues that may be particular about doing some of these studies, but I don't think they're insurmountable, and I'm very optimistic that this is something that is very possible to do.
And no doubt one of the concerns that will come if we are lucky enough to get to that point is cost, because such a new therapeutic, if successfully developed traditionally while it's still under patent, tends to be, at least if it's like other rare diseases in this country, exorbitantly expensive. Can you imagine how that kind of debate might work itself out in a positive way?
I think the answer with cost is really the lessons. Again, if you look at HIV, there are a lot of lessons that can be learned from what or how they have dealt with the issue of cost of drugs. And this is really an issue or something that if we strategically say, this is something that has to be done. It is a huge public health burden. There shouldn't be a debate about how expensive it is or not. It should be done, and we should find a way of engaging with industry, engaging with governments to make sure that this is made affordable to the people who need it. And if they've been able to do that with antiretroviral drugs, if they've been able to use it with antimalarials, I'm pretty optimistic, again, that this is something that can certainly be done. And the good thing about this is that this is a disease that is not just limited to Africa. So this is something that is relevant or pertinent outside of Africa as well, which would hopefully be able, would make industry see that this is something worth investing in.
Indeed.
Yeah.
Over here.
Yeah, hi. Hi, great presentation. I was recently at the International Stroke Conference, and there was a great talk by Dr. Robert Adams about the STOP trial, in which children with sickle cell disease are transfused with blood to prevent strokes. So I just wanted to ask you, how often do you see strokes in children with sickle cell disease in Africa, and do you use transfusion therapy?
So within the cohort in Tanzania and within groups that are working, stroke is common. So the prevalence of it is comparable to that in the reports that have been published from here and other places in Europe. So it is a problem, and we have set up facilities for being able to identify those with high TCD and who are therefore at high risk of getting stroke, and as well as the fact that there is an improvement in imaging. So we're able as well, to do CT scan and MRI to kind of characterize the type of stroke that patients have. The issue obviously is that blood transfusion is a challenge in our setting. And there has been, fortunately, huge investments in a lot of countries which have HIV in improving blood safety. So there's been improvement in the infrastructure as far as blood transfusion is concerned. And the issue or the challenge there is how we can improve blood supply by encouraging donors, etc. And so that capacity or that resource is growing, but we're still not yet in a position to be able to give or provide or offer patients the possibility of long-term blood transfusion programs. So what we do in our setting and other people, what they do is when patients come in with acute stroke, you transfuse as a form of treatment, and then if you can, you then would initiate patients to come in for a transfusion once a month, but this is not really sustainable. So a lot of times we're not able to continue doing that. And this is where for us hydroxyurea is the alternative. Because rather than just look at patients who you know have a high TCD and who know already have had a stroke, it's very difficult if hydroxyurea is there for you to say, well, it hasn't been proven to be very effective, the studies are not. So we do give patients hydroxyurea for secondary prevention.
You noted 4 different haplotypes. Are they believed to be traced to a single mutational event in HBS?
No, it's thought that they happened independently.
Which is fairly amazing that the same nucleotide could mutate to the same alternative nucleotide independently multiple times. But you can imagine with a very strong selection, and that basically every nucleotide in the genome is getting mutated on a regular basis, because we all carry about 60 mutations our parents didn't have. You can see how this could happen. It was a stunner when it was first realized that these were independent events, but they clearly are. Yes, over here.
Hi, Julie. I have a 2-part question. The first is as you think about the public health system in places like in Africa, and let's say using Tanzania as an example, what role do you think the traditional newborn screening versus point of care testing at the time of first illness, what are the relative roles that both will play in identifying sickle cell disease? And are there networks that are developing that would allow young patients, once identified, to be incorporated into healthcare and clinical research networks that would then make them accessible to some of the research that you're very interested in doing? Thank you.
So in terms of the second question, what a lot of us have done is to look and say, within the health system, integrate it into non-communicable diseases, because this is an initiative that is already ongoing, and it's easier to include sickle cell disease as a disease within that program, rather than say, well, this should be within HIV or as a chronic disease. So different countries have done different things, but by and large, most of us, what we have done within Africa is to say, make it part of the non-communicable disease strategy. So again, the approach there is to say, when we are going out to teach or to train individuals, or healthcare providers about NCDs, the aim is in areas where there's low prevalence of sickle cell disease, then we have one person who is an NCD person who provides care to everybody with diabetes, sickle, etc., because the prevalence of those conditions is low and maybe the resources are low. In areas where there's high prevalence of disease and the need is greater, then in those instances, NCD would need to automatically splinter into disease-specific services to cater for the demand that is there. And the health system within a lot of the African countries is geared towards working with this. Mainly, again, with the HIV, they've done a lot of work in making sure that there's healthcare at community level right up to the top. So again, when we go in, it's going to say, what have they done, what works, what doesn't work, and how can we use this person? Instead of there being one HIV person, one NCD, why don't we have one person who provides, you know, service for all. So this is something that I think is doable, and we're not starting from scratch. As far as newborn screening is concerned, I think because there's quite a lot of, there's high coverage as far as immunization is concerned, and a lot of, despite the fact that a lot of individuals give birth at home in the rural setting, but most of them by and large do within at least 5 days go to a health facility or have access to a health facility at least for immunization. So it means that as far as screening, newborn screening is concerned, we would be able to do that. And it makes sense. Again, this is something that we need to look at, and we have been looking at. Practically, it makes sense to screen, to collect the dried blood spots in the community, and then centralize the laboratories rather than, you know, have the point of care diagnostic there. But I don't think it's an either or. I think if we can improve the way that we do the diagnosis, then it's better. And point of care is something that would be very valuable. Yeah.
Over here.
Julie, that was a wonderful talk. I think it's worth emphasizing a point that you just made a moment ago about issues such as the practical limitations about chronic transfusion for your post-stroke patients and the use of hydroxyurea for secondary stroke prophylaxis, because this sort of issue represents actually a very much an opportunity of low-hanging fruit to take advantage of these situations where what looks like a disadvantage, a resource-limited situation, may actually provide a stimulus for developing new strategies. It may, in fact, be ultimately more advantageous than strategies such as chronic transfusion, whereas there may be an understandable reluctance in this country to launch a trial of other alternatives to what already seems to be a safe and effective alternative of chronic transfusion, but it may not necessarily be the best, or there may be a mutual benefit and ultimately a leadership position in research for these sorts of situations borne by your African network.
That's true. No, that's a good point. Thank you.
Call that reverse innovation. Yes, over here.
Hi, thank you for such an informative talk. I'm a graduate student here at the NIH, and I had a question about educating expecting parents who are both carriers of the sickle cell trait, and what impact or role you think that might play, because I know that environmental and nutritional factors also contribute a lot to the manifestation of this disease.
So this is, as far as sickle, and unfortunately, this is an issue that they have in places where they already are in a position not only to do counseling before families start thinking about children, but also with antenatal or prenatal testing. And the issue around sickle cell disease is that if you do have SS disease, there is no way of knowing early on that this is an individual who has severe disease. So it becomes very difficult to advise a couple who have 1 in 4 chance of getting a child with SS that that child will be severely affected. It becomes a lot easier when you know that a disease will result in severe disease, will result in severe consequences. But in this instance, you don't know whether this will be somebody with mild disease or with severe disease. And so I think this is something that we have to provide, and we have to give people options. And there's work that's been done by Ambrose Wonkam in Cameroon that has shown that within an African setting, the whole issue of prenatal diagnosis is not as, is not as unaccepted as one would have thought. So people are willing to kind of think about it and consider it, but it becomes difficult. What do you really advise somebody who has an SS child who is likely not to have severe disease? And you can't really predict that, yeah. But it's something that I think we need to think about, and we need to do qualitative social research to kind of find out what is the right way of doing things, and it will be different in every environment, yeah.
Last question, yes.
Hi. So I'm Nigerian, and for me, sickle cell is something that's not just a disease that's good for scientific research, but it's also a life-going thing. Like, I mean, I experience it every day with family members. I have a cousin who now is like in his late 40s, he's SS, and having to see his trials of like having crisis, and in elementary school, I have friends who die from sickle cell disease. And one of the things that I've always had issues with is in terms of how do you go about, because you mentioned advocacy as a way in which we should look at in Africa. And the common thinking, especially when it comes to sickle cell, is from the very start. You mentioned the case of, she mentioned the case of a couple in which both of them are sickle cell trait. That would be considered like forbidden. They shouldn't even get married in the first place, because that's what people would say. Try and stop it from the very beginning. Do not get married. There's this whole idea of pre-planning. So like, if you have a sickle cell trait, they would advise marry someone who doesn't have the disease trait. So I really want to know how you would go about with advocacy, how you would go about telling a lame person, a lame African person about all this information that we hear, that sickle cell disease is not just something that is a death sentence. There are severities in terms of just the disease itself, and also in terms of, sickle cell traits and all of that stuff. So how would you really go about with advocacy? Because when I talk with the information that I know about sickle cell, when I talk to my friends who are here and have learned a little bit of biology about sickle cell, they still have, the basic thinking that it's a death sentence, and from the very beginning, you shouldn't even have a child that has, that you shouldn't even give the probability of having a child that has sickle cell. So, yeah, that's my question.
I think it's very important, the advocacy, and making sure that the message, and again, one can assume, or one would assume, that everybody who has had a university education will know about sickle cell disease, but that's not the case. And when we talk about advocacy and information, it's all of us. There's assumptions that we may probably have, and so we need to make sure that everybody understands what are the risks and what are the benefits of having sickle cell trait. In terms of being able to advise, I think it's first of all, there are a lot of advocacy. There are not a lot, but there are an increasing number of advocacy groups all across Africa and globally as well. And there is a lot of information that is already there to answer those questions. We won't be starting from scratch. These questions that are pertinent, or that are asked in the US, are the same questions that are going to be asked in an African setting. So we're not starting from scratch. We have the answers, and it's just a matter of being able to translate them so that they're locally relevant. So the information is there, the capacity is there in terms of being able to provide information about chronic disease, about diseases that were considered to be fatal, again, and conditions that are considered to have a stigma attached to it because of HIV. They've dealt, fortunately, with all those issues, and they've been able to demonstrate how you can use advocacy measures and strategies to kind of debunk the whole mystery around diseases like sickle cell disease or HIV. So we're not starting from scratch. There are resources from different diseases. There are resources from sickle cell disease in other populations, and we will be able to change the understanding of the disease and inform people better.
Well, I want to thank all of you for excellent questions and a really nice discussion, a wonderful presentation. There's going to be a reception in the NIH library to which you're all cordially invited if you wish to continue the conversation. And please join me then in thanking again our speaker, Dr. Julie McConney.